Hello, everyone, and welcome to the 17th episode of my publication at a glance. Today, we have the pleasure to welcome Marc-Antoine Panosetti, who completed his PhD at the University Côte d'Azur in Nice, and who is currently a postdoc researcher at the University of Oxford. Today, he will share results about breast cancer cells. And Marc Panosetti, the floor is yours.
Thank you very much, Jerome, for the nice introduction. And so, of course, to the whole young research fellows of the SCT for giving me this opportunity to present this work that I've been doing during my PhD in collaboration with the University Côte d'Azur and the Italian Institute of Technology in Genoa. So a bit of background to start, but I think the audience all know about this, but RNA as a biological target is a growing focus of academia and pharma industry recently, which led to identification of several successful drugs, most important one being risdiplam for spinal muscular atrophy, SMA. Why is there a growing focus about that? Of course, because now we are quite well aware that 1.5% of our whole genome is encoding proteins, and the rest is encoding RNA, so at least 70% of it is encoding non-coding RNA. This was what we were interested in the lab in Nice, where we were most specifically interested in microRNAs. So microRNAs are very small non-coding RNAs, which are responsible for the regulation of gene expression. And they are now, their role in many human diseases is well known, usually most importantly negatively. So to make a long story short, microRNAs are able to recognize target messenger RNA, bind to them, and prevent their translation into proteins. And this is where we wanted to have a role to play to their maturation. So microRNA comes from a precursor called pre-microRNA, a longer sequence that is usually cleaved by an enzyme called dicer. It cuts it and makes a shorter version, the mature microRNA, that will recognize the messenger RNA. And the idea of the project was to develop small molecules that could bind to the pre-microRNA and prevent the enzyme to cut it through. So to do so, of course, we chose a target, microRNA 210, for its well-studied overexpression role in cancer, and more specifically in hypoxic conditions, so low oxygen levels in cancer cells, which are the most complicated region of cancer to target. And this target needed new chemical probes to elucidate its real potential as therapeutic target. So we started to look at what was known in the literature. There was only one compound, this one, target Premier 210, which was known for its affinity, but a bit more complicated on the selectivity. And this is why we started with a bit of computational to actually come up with a new series of our own. And we came up with this tristiazole scaffold, which brings me to the SAR of the paper. So for the sake of time, all the chemistry, I'm going to have to ask you to read my paper to look at it. But then, so we start by modifying the right-hand side of the scaffold by introducing different lateral chain, including the one that's already published. And by different fluorescent assays, we tested them for affinity and inhibition towards the enzyme, which we could select this one with low micromolar activity. And we decided to modify all of its left-hand side, introducing different moieties. The idea being to have a variety of moieties and to have a collaborative effect with all the different parts of the molecules to obtain the best inhibitor. So all of these were also tested by myself, same for affinity and inhibition, where we could make up some SAR conclusions. So for this target, it seems to be that heteroaromatic rings were not very suitable for affinity, and we were able to identify 3 promising compounds with low micromolar KD and low micromolar IC50s, all bearing cyclic amines or polar motif, which seems to be very essential for activity. And with this, I moved to more in cells biology, and I changed lab. So first of all, the first thing that we had to check was that microRNA 210 was overexpressed in hypoxic conditions. And if you look at this middle graph, we use a very well-known in the biology field compound called cobalt chloride that induces hypoxic conditions in cancer cells. So you can see here the overexpression compared to control of the microRNA. And with one of my best compounds, we were able to see the reversion of its overexpression. It was seen on the microRNA level. It was also seen on the pre-microRNA level. And then we're quite happy to look at well-known hypoxic markers, one of the most famous one being CA9, which is always overexpressed in hypoxia, as you can see here in control. And with the best compounds that we have, we reverted this. And with other two compounds also, we're quite happy to have some potential backup compounds, of course, for the future of the project. And so this is what brings me a bit to the conclusion about the fact that we were able to identify this compound, 6A, which was able to revert the levels of microRNA and to revert the hypoxic condition in those triple negative breast cancer cell lines, which is something quite promising for example, development of a compound or new chemotherapy, because of course, hypoxic condition being the most complicated one to target in cancer cell, if you can revert the zones of the cancer to normal condition, it'd be easier to target later on. And on that, thank you very much for listening.
Thank you very much for this very nice snapshot of that research. I will encourage, of course, everyone watching to check your paper. And if anyone is interested in this kind of format to present their research, their own research, PhD or postdoc researcher, they can just contact us via the link that are on that slide. Thank you very much and see you next time.